Now that we are familiar with some basic anatomy of skeletal muscles, the question we will be addressing in this segment is how do skeletal muscles generate forces? So the answer to this question is that there are structures within the muscle cell called sarcomeres, and these sarcomeres shorten to generate forces. But we haven't looked at a sarcomere yet. So what is a sarcomere? So recall that we had muscle cells, which were also referred to as muscle fibers. And these fibers are packed with rod-like organelles called myofibrils. Now, if we take out a single myofibril from the skeletal muscle cell, we see that the myofibril is composed of thousands of smaller repeating subunits that are linked end to end. And these are the subunits that we call sarcomeres. So it turns out that one sarcomere actually sits between two vertical bands in the myofibril called Z-discs. And so what we're seeing here is we're getting a closer look at the myofibril that we've pulled out of this skeletal muscle cell here. And we see that there are overlapping structures, overlapping lines within this sarcomere here. We'll take a look at those in a second, but what you're seeing in this diagram here is that you're seeing these very thin vertical lines here, and these mark the ends of the sarcomere here, and these ends are called the Z-discs, and so one single sarcomere sits between these two Z-discs here. And what you're seeing on either side is part of another sarcomere, so we can't see the other Z-discs over here because they've been cut off of this diagram here, but what you would see is you would see more Z-discs that would mark the end of these sarcomeres on the right and left side of this one. So the sarcomeres are linked end to end, and the Z-discs are the end of one sarcomere and the beginning of another sarcomere. So let's look at a more simplified diagram of a sarcomere. So what you're seeing here are the Z-discs and the overlapping protein filaments that are inside of the sarcomere between the two Z-discs here. So in this diagram, at the top, we have a sarcomere in a muscle that is relaxed, and at the bottom of the diagram, we have a sarcomere in a muscle that has contracted. Now notice really the major difference between the relaxed and the contracted diagrams here is that the Z-discs are much closer together, that is, these purple vertical lines here are much closer together in the contracted muscle, and they're much further apart in the relaxed muscle. So when the muscle contracts, the Z-discs of the sarcomeres are getting close together, which means that the distance between the two Z-discs is getting shorter. And so that's what's actually shortening when the skeletal muscle contracts. So this process requires two things. It requires ATP and calcium. So if one of these substances is not present at the same time as the other, the muscle will not contract properly. So the structure of sarcomeres is made possible by, or the overlapping structures within the center of the sarcomere is possible because the sarcomeres are constructed from these muscle filaments that are called myofilaments. And so there are three types of myofilaments that we're going to take a look at. We're going to take a look at the thin myofilaments. They're also just called thin filaments, the thick myofilaments or the thick filaments, and the elastic myofilaments. So those are the three major filaments that we see that make up our sarcomere here between our two Z-discs. So let's start by looking at the thin filaments first. So the thin filaments, what you're seeing here, are constructed from these blue bead-like proteins in this diagram here. They're not actually blue in real life, but they are kind of a bead-shaped structure, and these bead-like proteins are called actin. And so these actin proteins form strands, and the thin filament actually consists of two strands of these actin proteins that are twisted together, kind of like beads on a string. And also associated with the thin filament we see are two regulatory proteins in the yellowish colors here. And these two regulatory proteins, one of them is called tropomyosin, which is this thread-like protein, and the tropomyosin is supported by another protein called troponin. Now, these two regulatory proteins, that is tropomyosin and troponin, prevent the muscle from contracting when the muscle is not stimulated. And so we'll see where these come into play a little bit later. So we've got the thin myofilament, or the thin filament, which is constructed from two strands of actin proteins that are twisted together. And we also see tropomyosin and troponin, which are regulatory proteins that are associated with that thin myofilament. So that is the thin myofilament there. The thin myofilaments are anchored to the Z discs on either side of the sarcomere here. So we see the thin myofilament here, and it is secured to the Z disc, and the remainder of the thin myofilament extends away from the Z disc towards the center of the sarcomere. And so we see these thin myofilaments anchored to the Z discs and extending inward towards the center of our sarcomere here. So let's take a look at the thick myofilament now. These thick myofilaments are bundles that are made of proteins called myosin. Now, a single myosin molecule essentially looks like two golf clubs that have had their handles twisted together. The part of the myosin here that is twisted together with the other myosin molecule, these are the tails of the myosin. Towards this little head-like region here, we have this flexible hinge region that's in close proximity to these club-like heads. The heads, you'll see, have two binding sites. One of those binding sites is for ATP, and ATP is essential in not necessarily helping the myosin flex at the hinge region here, but the ATP is actually important for resetting the muscle so that it can contract again. And so we'll talk about the role of ATP a little bit later in muscle contraction. But basically what ATP does is it helps the myosin unbind itself or become detached from the actin after it has contracted. And so ATP is really important for breaking the contact between the myosin and the actin so that the muscle can, the myosin molecules can contract again and do another pull for the muscle. So we see an ATP binding site on the head region for the myosin molecule. There's also an actin binding site because the myosin binds to the actin and pulls, and so that's where the myosin will actually grab onto the actin thin filament and pull, and that's ultimately what causes the sarcomere to shorten. So the thick myofilaments are actually suspended from the center of the sarcomere here, and we see that their little club-like heads are extending in many different directions towards the thin filaments that are extending out from the Z discs and towards the center of that sarcomere there. Now the third myofilament is this elastic myofilament, and the elastic myofilament is made of a large protein called titin, and so that's why it's called titin, because the word titin actually means like large, and so this is a very large protein that kind of superficially it looks as though it is anchored to the Z disc, which it is. It has this little kind of coiled like spring region here, and then we kind of see that it attaches itself to the thick myofilament here. But what actually happened, or what actually is going on here, is that the elastic filament actually extends through the core, through the middle of the thick myofilament, and extends pretty close to the center of the sarcomere. So one elastic filament is about half of a sarcomere in length, and it's just one protein, so it's a very big protein, which is why it is referred to as titin. So the elastic myofilament does a little bit more than just suspending the thick myofilament from the center of the sarcomere. So near the Z disc here, the elastic myofilament, as I mentioned earlier, kind of looks a little bit like a spring, and that's because the elastic myofilament helps give the muscle its spring-like and its elastic-like properties. So that is, the muscle can be stretched out to a longer length, and then after the force that stretched that muscle out is released, the muscle will return back to its resting length. And so for that reason, muscle has some spring-like properties, and part of that spring-like property comes from the elastic filaments that are within the sarcomere. So those are the three basic myofibrils of myofilaments, not myofibrils, of the sarcomere. So if we were to look at a light micrograph image of an actual sarcomere, you would see these two distinct bands on either side of the Z-disc here, I should say, between the two Z-discs here. The Z-discs are these dark, thin vertical lines in our diagram here, and they're pretty closely lined up here with our image graphic of a sarcomere here. So this dark band that you see in the middle of the sarcomere is called the A band, and I always think it's the dark band since dark has an A in it. So this is the dark band within the sarcomere. And the reason that it's dark is because in the center of the sarcomere is where we have overlap of the thick filaments with the thin filaments. And so because there's a lot of material packed in there, this looks as though it is very dense and very dark in the center of the sarcomere. Whereas out towards the periphery of the sarcomere, we have these light-colored bands that are on each side of the Z-disc, and these are the light bands, or the I bands. And I always think or I band is associated with the light band, since light has an I in it. And so what we're seeing here on either side of the Z-disc and the I bands here is we've got the thin filaments, we've got the elastic filaments, so we can clearly see that there's some material here, but it's not as densely packed as the A band is, and so it doesn't appear as dark in the microscope image. And so for that reason, we get these two slightly lighter colored I bands on either side of the Z-disc here, and in the very center of the sarcomere, we have the dark A bands, which represent the overlap between the thick and the thin myofilaments. So now that we have seen the components of the sarcomere, how do we put all that together? What actually happens when the muscles contract? So it turns out, as I mentioned, that the myosin that's found in the thick filaments, the heads of the myosin molecules attach themselves to the actin of the thin filaments, and then that myosin molecule pulls. And since the thin filaments are attached to the Z-disc, the sarcomere gets shorter when the myosin pulls on the actin. And so the name for this phenomenon is the sliding filament model, due to the fact that these actin filaments are sliding over the thick filaments as the thick filaments are pulling on them. So notice here too that the thick and the thin filaments did not get shorter during contraction. So the only thing that got shorter was the distance between the two Z-discs in the sarcomere. So when we talk about muscle shortening, we're really talking about how shorter are the sarcomeres getting when that muscle contracts. And so that's what's really driving the shortening of the muscle.